Okay, so moving on with the next part of the topic in this lecture on cell cycle. So we will look at cancer. So cancer cells are defined by two heritable properties. So first is that they reproduce in defiance of the normal restraints on cell growth and division. So that means the normal switches are not really functioning, or rather they are functioning far too well. They are not turning off. And they invade and colonize territories normally reserved for other cells. So just a brief look into the different definitions into cancer. So there are many different types of cancer because we have many different types of cells in our body. And there are many different terminologies that might confuse you. So let's look at some of the terminologies. So when we say neoplasm, it means new growth. It is a tumor. It arises from the abnormal cell that grows. When we say it grows, it increases in mass. And it proliferates, that means it multiplies, or basically it undergoes cell division, out of control. So an out of control mass of cell that is growing. So that's your tumor, or the more technical term is neoplasm. So when you have a benign, these are neoplastic cells, tumor cells, that have yet to become invasive. They are not yet invading other parts of the body. When they become malignant, that is where you have a true cancer. So this happens when the tumor cells, your neoplasm, have acquired the ability to invade the surrounding tissue. Then we have metastasis. These are secondary tumors arising from malignant cells that broke loose. They enter the blood or the lymphatic vessels and colonize other parts of the body. We have carcinomas. These are cancers arising from epithelial cells. For the sarcomas, these arise from connective tissues or muscle cells. And then the ones that not included, usually these are the leukemias and the lymphomas. These are derived from white blood cells and their precursors, the hemopoietic cells. So these are just a few of the very large number, a myriad of different terms when we talk about cancer. But again, we are just going to discuss cancer in the context of the cell cycle here, not really discussing the cancer as, because it's really a field of its own. Okay, so just a brief look, what is a normal versus benign versus your malignant. So this is your normal on the leftmost part. This is your normal cell. So usually it's say for example a colon cancer, for instance. You have your normal duct, so there's a movement here. And then if you have a tumor, the tumor is growing. So if you can see, although it's benign, it's not invading the surrounding cells, but it's constricting the space, the lumen of the normal duct. And then when you have malignant tumor, uh, actually in your tumor, basically, or in this mass of cells, there is a combination of the malignant cancer cells and the benign cancer cells. So this is a tumor that has gone malignant and they have basically metastasized. So here are cells that have broken loose and are starting to invade other parts of the body. So this is your malignant cells, the malignant tumors. So they have invaded, they are starting to invade other cells or even the surrounding cells. So basically, that is the oversimplified. This is basically the oversimplified view of the normal versus benign versus malignant cancer cells. Okay, so what causes cancer? So there are when you look at the genetic of the genes, because again the cancers, or rather the switches, the cell cycle switches, they usually act upon the genes. And since the switches are usually not usually, they are proteins, they are actually products of genes. So at the base of it, at the heart of it, is the genes. So we look at the two types of mutation that can cause cancer. So we have, these are the cancer critical mutations. And there are two categories, the overactivity and the underactivity mutation. So for overactivity mutation, we call it the gain of function mutation. So for example, your normal cell, a single mutation event creates the oncogene. So all of us have a proto-oncogene. It's called a proto-oncogene because it has the potential to become the oncogene. And why do we have those proto-oncogenes? We have those proto-oncogenes because more often than not, or rather, most of the proto-oncogenes, the functions of these genes is actually to signal it's the go signal for the cell cycle. It usually is the signal. They encode for proteins that act on the signal that says we will proceed from the G1 phase to the S phase, or we will proceed from the S to the G2, or we will proceed from the G2 to the M phase. So these are genes that signals the cell to divide, to replicate their DNA and to divide. And a single mutation of this is the gain of function. It promotes the transformation of the cell to become tumorigenic. Basically, because why only one? It needs only one because it just needs a go signal for the cell to only need, actually you basically just need one go signal, or at least for this type of oncogene, for the product of this oncogene, you just need one signal from this one to say that we need to proceed to the cell division. So basically that's the gain of function mutation. Only one is needed. And this is one of the reason why, for example, BRCA1. BRCA1 is one of the most popular and most commonly studied, most well-known oncogene, which is responsible for breast cancer. And you all know that breast cancer is a heritable type of if you have a family member, your mother or grandmother have breast cancer, you are almost certain to have breast cancer yourself. That is because if you have inherited the oncogene from your mother, only one mutated oncogene, you are already at risk for cancer. I'm not saying that you already have the cancer, but you are already at risk. And it's a very, very high chance that you will develop cancer later on. So basically, and that is because of this single gain of function mutation. The other one is the loss of function mutation, and this is seen on tumor suppressor genes. Okay? So in this one, you must have two mutations, two loss of function mutations, because these tumor suppressor genes are always present in our cells. They stop, basically, it's a tumor suppressor because they stop the progress of the cell cycle. They stop cell division. They basically function the opposite way of the oncogenes. They are the ones that say, no, we are not proceeding there, stop. But if the one that is stopping them is gone, both of them, so we need two mutations for them to be really to really have cancer, because again we have two copies. If we lost one copy, we have a backup, and it's enough. It's basically enough to have one the backup to actually to function. But if even the backup is gone, you have nobody will tell the cell to stop. There is no signal anymore that will tell the cell to stop. You are going to become a cancer cell. No more. So basically, you need to have these two mutations for it to go. And usually, the loss of function of the tumor suppressor genes is actually much more critical than the gain of function of a single oncogene, because the single oncogene will tell you to go, but if you still have this tumor suppressor to tell you to stop, to tell the cell to stop, there's still some sort of control somehow. But if you have this one and then you lost your tumor suppressor gene, both copies, you basically have nothing else to stop your cancer, but except for external forces. But for the internal control of the cell has totally lost control of its cell cycle. So basically, these are the two types of mutation you have: the gain of function of the oncogene and the loss of function of your tumor suppressor genes. And that is why you have this dominant and recessive types of cancer. So the dominant one is for the gain of function mutation. So breast cancer is more or less a dominant type of cancer because you only need one to have the risk to potentially have the cancer. Whereas the loss of function mutations, this is recessive because you need to lose two of them for you to completely lose the function of your tumor suppressor gene. So basically, that's the two different types of mutations. And mind you, a cell that has a neoplastic cell, basically the tumor cells, since they tend to multiply fast, they acquire lots and lots and lots of mutation. And among those mutation, you will have multiple oncogenes, rather proto-oncogenes becoming oncogenes, and multiple tumor suppressor genes that are losing their functions. Okay, so one of the when I told you about the tumor suppressor genes, and one of the most critical of the tumor suppressor genes, one of the most important of them, is called the P53. P53 is a protein, a signaling protein. It's a tumor suppressor protein. So what does your P53 do? So if you have your if you have hyperproliferative signal, so you have a signal that this cell is this mass of cell are proliferating, but they should not be proliferating. It will tell the P53 to stop the cell cycle, to have senescence or apoptosis. Senescence is basically the cell, although it has multiplied, it has replicated its DNA, but it won't undergo cell division. It will just go into a dormant stage, senescent stage, or it will just commit suicide, which is apoptosis. So if you have these signals, hyperproliferative signal, DNA damage, telomere shortening, and even hypoxia. Hypoxia is the loss of oxygen. And this is usually a signal that says there are too many cells in this area. So much so that the oxygen supply has become too depleted. There are too much demand versus the supply. And that means you have overcrowded cells in that area, and it will tell your P53 that something is wrong. You should arrest the cell cycle. You should either undergo senescence or should undergo apoptosis. So the P53 has it's actually at the center of one of the many different signals in the cell cycle signals and the cancer metabolic signals. And the loss of the P53 function is actually one of the things that they are looking for when they are diagnosing the stages of cancer. It's usually found at the end stage of the cancer, the higher stages, stage 4, and so on. The loss of this P53, because this is very critical. It's very important. If you look at studies, looking at cancer studies, different, even though breast cancer, colon cancer, even different types of cancers, P53 is still at the center point of the signal, the tumor suppressing signal among those cancer. So this is just a tip of the iceberg, and we are just looking at cancer in terms of the cell cycle controls. But basically, cancer is the outcome when your normal cell cycle control becomes disrupted. Okay, but again, cancer is a field of study that is very wide. There's many different things going on there, and it's still under study. We still do not know fully what is going on. Okay, so this is just a tip of the iceberg. The rest of them is for you to find out. Okay, and that ends this lecture. Have a nice day.